Hello everyone, welcome back to Dr. K. Primer video lecture series presenting by Dr. K. Prem Sagar. That's me. In today's lecture, we'll talk about cDNA library. So today's lecture, that is cDNA library, is divided into the under 9 headings. The first one is we'll define the what is cDNA and cDNA library, and why we have to construct this cDNA library, need of cDNA library construction, and what are the types of cDNA libraries, and how do you construct, what are the steps involved in the cDNA library construction, and what are the vectors and methods used in the cDNA library construction. How do you screen? What are the methods available to screen the your clone of interest from the cDNA library? What are the applications of cDNA library? And what are the disadvantages of a regular cDNA library? And finally, we'll take a small test to test your learning, whatever you learn, right? So we'll go to the next slide and define what is cDNA. What is cDNA? So you all know that retroviruses have an enzyme that's called as reverse transcriptase, which can convert its mRNA into DNA, that DNA integrated into the host chromosomes. Commercially available reverse transcriptases, such as AME reverse transcriptase or MMLV, Mullally-Murine leukemia virus or avian myoblasts reverse transcriptases, can use the mRNA, and converts into the cDNA, converts into the DNA, right? Throughout the process called as reverse transcription. Reverse transcriptases catalyzes the conversion of mRNA into DNA. That process is called as reverse transcription. The DNA which is synthesized in this process is complementary to the initial mRNA. That's why the DNA is called as cDNA. You can see reverse transcriptase catalyzes the in vitro synthesis of DNA by using mRNA as a template. That process is called as reverse transcription. The DNA is complementary to the initial mRNA, hence that is termed as cDNA. You can see here, this is a eukaryotic mRNA, which is having the poly A tail, which is incubated with the oligo DT primer and reverse transcriptase and DNTPs. The oligo DT primer will anneal to the poly A tail of mRNA. Then the reverse transcriptase will bind to the primer and mRNA complex, and it polymerizes the or synthesizes the DNA by using DNTPs. You can see this is called as a cDNA. mRNA is converted into the DNA, right, by reverse transcriptase. This process is called as reverse transcription. The DNA is called as cDNA. The cDNA is complementary to the initial mRNA. Now we will see what is a cDNA library. So the cDNA you prepared by using the mRNA molecules, they are inserted into the suitable vectors and transferred to the host cells. And you screen the cells for a, for those cDNA present or absent. That is called as a cDNA library. You can see here, a cDNA library is a combination of cloned cDNA fragments constituting some portion of the transcriptome of organism, which are inserted into a number of host cells. cDNA library is a combination of cloned cDNA fragments constituting of some portion of the transcriptome, which are inserted into the number of host cells. mRNA is isolated, converted into cDNA. That cDNA is inserted into the suitable vector, and those vectors are transformed into the E. coli cells. Now these transformants are screened for the presence or absence of the cDNA. So those who the positives collect the positive clones which are having the cDNA molecules are collectively called as a cDNA library. One more definition here. A collection of recombinant clones consists of cDNA of mRNA molecules expressed by an organism or tissue or a cell under particular condition. Here you say recombinant clones consist of a cDNA of mRNA molecules expressed by an organism. So here I'm saying again that mRNA is isolated and it is converted into the cDNA and put into the vectors. Our vectors are transformed and screened for the presence of cDNA in the vector system. So if you say, whatever the positive clones or recombinant molecules, that collection of recombinant clones which consist of a cDNA of mRNA molecule expressed by the tissue or cell under a particular condition, that's what is called as a cDNA library. Why we have to construct the cDNA library? What is the need of cDNA library construction? Because tissue or a cell or an organism, they'll express the certain number of genes all the time. They don't express all the genes at a time, right? So cell or a organism or a tissue, if you say, they have a unique population of mRNA molecule. Each cell or a tissue or an organism have a unique population of mRNA molecules, right? So that the unique population and degree of expression of particular gene is varies from one cell to other cell and one condition to other condition. You see, each organism, tissue or cell type has a unique population of mRNA molecule. When you change the condition also, the degree of expression of particular gene or genes which are expressed in that particular condition varies from one condition to other condition to cope up or to adapt to that particular condition. So here, what I'm trying to tell you all that any cell expresses a set of genes and those mRNAs are converted to proteins so that that cell can survive or thrive in a particular condition. Whatever genes which are required to thrive or tolerate or adapt to the environment, they'll be expressed, right? So when you change the condition, when you expose the cell to very adverse condition, automatically the population of mRNA varies. That's why to understand the role of genes which are responsible for tolerance or adaptations or stresses, we have to isolate mRNA. But problem is, mRNA is not at all stable one. You can't clone it. You can't amplify it. We have to convert it a stable form. That's a cDNA. So once you convert into the cDNA of particular condition, and if you clone into the clone and you get a positive or recombinant molecule, recombinant clones, that is called as a cDNA library. You can see here, not all genes are expressed at the same time, as I told you. So expressed genes means mRNA molecules, they're unstable. You cannot store them for a very long time, or you cannot clone them, or you cannot amplify them. What you have to do is, just you have to convert the mRNA from particular situation, or a particular cell, or a particular tissue, or a particular stress condition. You convert them into a cDNA. And this converted cDNA molecules are cloned. Then you got a library. So when you have a cDNA library of a particular cell under particular condition, you can sequence all the clones, and you will be knowing what are the genes expressed in that particular condition. What are the genes which are required for that particular condition, you will be knowing. Right? So mRNA converted to cDNA, cDNA is cloned, all cDNA molecules are cloned, are sequenced. Then you will have information about cell machinery, or what is happening in the cell in a particular condition. Yeah, as I told you, that stable form of mRNA is a cDNA, right? This is the need of cDNA library. What are the types of cDNA libraries? You all know a regular cDNA library. That's our first one. Regular or normal cDNA library. And the second one is normalized cDNA library. Here, some genes are highly expressed, some genes are poorly expressed. So when you convert that sample into cDNA, the more cDNA belongs to 1 or 2 genes, and the more clones belongs to 1 or 2 genes. So it is the regular cDNA library is not a representative of every gene expressed under that condition. So that's why we have to normalize, means you have to remove the more prevalent mRNA molecules. Then there is a scope to include the poorly or less expressed genes. That is called as a normalized cDNA library. We'll talk about this later. And subtractive cDNA library. One cell, if you keep in 2 different normal condition, abnormal condition, right? Normal condition, the genes which are expressed are different, and abnormal condition, genes which are expressed are different. So whatever is common between them can be separated, and whatever is uncommon between them can be separated, and that uncommon fraction is responsible for stress condition, stress tolerance, or adaptation. There you can get the subtractive cDNA library. So today we will focus on normal or regular cDNA library construction. Later on, we'll talk about normalized cDNA libraries and subtractive cDNA libraries. What are the steps involved in the construction of cDNA library? We'll see. There are 5 to 6 steps are there. The first one is, first thing is that isolation of total RNA or mRNA from the total RNA. You have to separate. You can separate. You need not. If you don't want, you need not to separate. And the second step is synthesis of first and second strand of cDNA. So mRNA isolation and convert into the first strand cDNA and followed by the second strand cDNA. And the third step is cDNA molecules are blunt. They don't have any cohesive ends. So you cannot clone them directly into any vector. What you have to do is, you have to add cohesive ends to the cDNA molecule. That is through here you can see You can use the linkers, adapters, or homopolymers. You can use the linkers, adapters, and homopolymers to the cDNA molecule so that they'll have cohesive ends. Then the fourth one is incorporation of cDNA into suitable vector. So you have to select the vector here. What is the best vector? Which is, the choice of the vector is all depends on the investigator. What do you want to do it, right? So vector, if you can take plasmids or cosmids, or you can take the lambda phase vectors or M13 vectors. You can take it, and you can insert the cDNA fragments into the vector. Once you ligated the cDNA into the vector, then if it is a plasmid, you will do the transformation. If the vector is a lambda phase vector, then you do the transfection. Then the sixth screening of cDNA library. So you have to screen for the positive clones, right? So these are the 5 to 6 steps which are involved in the construction of cDNA library. In the next slide, we will see the same thing in the picture manner, where I drawn this picture, entire picture. You can see one step by step. I can use the animation. So this is the genome. Genome means all sets of genes, right? A set of genes is called as genome, right? So it's a one particular cell genome, and it consists of genes A, B. For example, you say A, B, C, D, E, F, G, H, I, so on. These are the genes, right? So genes transcribe to give mRNA molecule, right? So you can see the transcription. That's a RNA polymerase. You see transcription. One more. Yeah. mRNA molecule. mRNA molecule. Here you can see E gene is transcribed. You get a mRNA molecule, right? So I'm going to tell you again and again, not all genes are expressed at the same time. A few genes are expressed. What gene is expressed, that depends on the cells which are there. Interaction with the environment will determine what genes to be expressed, what genes to be silent. Here, in this condition, A gene is expressed, and the E gene is expressed, and the I gene is expressed, and the N gene is expressed. So this is a RNA, right? So RNA and mRNA, and these are the mRNA molecules. So mRNA molecules are incubated with the oligo DT primers or random primers and DNTPs and the reverse transcriptase. So mRNA molecules which are isolated from this condition are incubated with the oligo DT primers, random primers, and DNTPs and reverse transcriptase. So what will happen? Oligo DT primers will bind to the poly A tail of mRNA, right? And random primers can bind wherever they are complementary, they are the hexamers. Once the primers are bind to the mRNA, reverse transcriptase catalyzes the synthesis of, copies the mRNA into DNA molecule. You see? You get this is what is this is the first strand of cDNA synthesis. Right? Once you have the first strand, you can use the clean-out DNA fragment or equal DNA polymerase or any other DNA polymerase to amplify second strand from the first strand, right? You get a second strand, right? Double strand cDNA. Now we got we have isolated mRNA and converted into the single strand and first strand. There are the two steps: isolation of RNA or mRNA. That's the first step, and the second one is conversion of mRNA and mRNA to cDNA. That's the first strand and second strand. That's the second step, right? Yeah. So we have completed two steps. One is first we isolated the mRNA, then converted to first strand and cDNA, first strand and second strand cDNA synthesis by using the reverse transcriptase and other DNA polymerases. Now you have cDNA molecules, but you see the cDNA molecules are do not possess a compatible cohesive ends. Whenever you want to clone any DNA fragment into the any vector, they should have a cohesive ends. When they have a compatible cohesive ends, they can ligate. Then they form a recombinant molecule. So the prepared double-strand cDNA molecules do not have compatible cohesive ends. So what we have to do is we have to add linkers or adapters or homopolymering so that the cohesive ends are added at the ends of a double-strand cDNA molecule. You see, these are the, you can say adapters or linkers or homopolymer tailing to the ends of a double-strand cDNA molecule. You can see here, linkers or adapters or homopolymer tailing is done to the double-strand cDNA molecule so that they are ligatable with the vector molecule. So now the next step is linearized vector. You can use the plasmid vectors or you can use the lambda phase based, they are insertional vectors or replacement vectors. So that depends on the choice of the investigator. Here, the vector is linearized and the linearized vector have a compatible ends. So you see, this is one more vector, right? So now you have a cDNA molecule, double-stranded cDNA molecule with the cohesive ends, and the vectors also have cohesive ends. The next step is ligation. This is the fourth step. The first step, you have isolation of RNA and the conversion into the first strand and second strand cDNA synthesis. And third one is addition of linkers and adapters to the cDNA molecule. And the fifth one is, linking, incorporation of cDNA into the vector, right? So here, linear vector with the compatible ends, ligate them, ligation of vector and cDNA with the help of T4 DNA ligase. Once the ligation is over, the ligation mixture is transformed into the E. coli cells if the vector is plasmid. If the vector is lambda phase based insertional vector or replacement vector, we have to do the transfection. Transfection or transformation. You can see transformation or transfection. Once you do the transformation and transfection, you get the colonies or plex, right? Then you have to do screening. See, screening. So now you see the 4 mRNA molecules you have in the beginning. They are converted to double-strand cDNA and they're cloned into the or incorporated into the vector. Now you see the 4 different clones, 4 different cells are having the 4 different recombinant plasmids with 4 different cDNA molecule, right? Yes or no? You see, cDNA library, a collection of recombinant clones consists of cDNA of mRNA molecules expressed by the organism. You see here, this is a A gene cDNA, this is a D D gene cDNA double strand, and this is J gene, and this is a N gene. So here, so you know the 4 clones which are representing the mRNA molecules expressed by the organism. So these 4 clones are called as cDNA library. You see, the collection of recombinant clones consists of a cDNA of mRNA molecules expressed by organism. You understand what is cDNA? mRNA molecules are expressed and they're converted to cDNA. They're incorporated into the vector and they're injected into the organism. So the positive organism or positive clones which are having the cDNA of mRNA molecule expressed. Those positives are recombinant clones which are representing the cDNA of mRNA expressed by the organism. This collection of recombinants are called as cDNA library. Yeah. This is the whole process, right? That's a, then I'm going to talk about what are the common vectors which are used in the cDNA library. You can see here, PBR322, there's a plasmid, M13 MP8, this is a M13 based vector where you can, once you clone into this vector, you can get the cloned fragment in a single strand form or double strand form. Single strand forms are very much required for DNA sequencing in those days. And lambda ZT10, that's a insertional vector and insertional vector and replacement vectors. So you can use the plasmids or M13 vectors or lambda phase based vectors. And what are the common methods used for a cDNA library construction? There are 2 methods. They are very famous. It's a Okayama and Berg method, and the Hedecker and Messing. These are the different methods available for cDNA library construction. How do you screen cDNA library? That's an interesting question, right? So unless you have some known sequence, you cannot get the whatever clone you want from that library, right? So there are 4 or 5 different methods are available for screening of cDNA library. The first one is using DNA probe with homologous sequence. For example, related species. Suppose a chimpanzee or gorilla and chimpanzees, humans and monkeys are related species. They share 98% similarity in the genome. Suppose a sequence, human genome is completely sequenced, right? So you can use human genes to screen the gorilla or chimpanzee cDNA library, right? Suppose cytochrome C oxidase from the human, you can use that gene as a probe to screen the cytochrome C oxidase from the chimpanzee or gorilla, because we are sharing almost 98% of similarity, right? And the second one is, you know the protein. If you know the amino acid sequence of protein, right? Suppose so for example, for example, some fungi, they have produced a lot of cell lysis, right? So the cell lysis amino acid sequence is determined. Once you know the protein's amino acid sequence, you can go back to the DNA, right? DNA to mRNA, mRNA to protein, right? If you know the protein sequence, that's amino acid sequence, we can convert that into the amino acid by using the triplet codon, you can convert it to the DNA, right? That's called as reverse genetics. If you know the protein amino acid sequence, we can prepare a probe to screen that particular gene of interest is there in the genomic cDNA library or not. You see, using you can use the amino sequence of the protein or antibody. If you have a protein and you raise the antibodies that, right? So when you clone your that particular cDNA into the expression vector, that expression vector produces the protein, right? So you already know that protein have antibody. So you go for the Western blotting and you can screen the gene of interest. Plus, minus, or differential screening can be done. Here I would drawn some diagrams for that. This is a cDNA library. You say there are the different clones. You see, chimpanzees and humans share the 98.9% of similar DNA. Suppose we know a genome sequence is completely known from the humans. So suppose you constructed a cDNA library of a chimpanzee, you can take genes from the human and to pick up whatever gene you want from the cDNA library, right? They're very similar. No, they're related one, right? Right? Homologous probes can be used to screen the cDNA library of related species. As I told you that if you have a protein of a particular organism, then you can raise the antibody. That antibody can be used to screen the clone of your interest through the Western blotting. Or if you have protein, you can go back to if you know the sequence of amino acid, that protein, then you can convert into the DNA, like right? So you see here methionine, and it codes with ATG, and this is a glutamic acid, GAA, and this is a serine, AGT. You can use the amino acid sequence to get a cDNA of that, right? That cDNA can be used as a probe here, right? So you can use the homologous probes to screen the cDNA library, or antibodies to screen the cDNA libraries, or you can use the translated cDNA as a probe to screen the cDNA library. See? So this is a identified, and this is what is a positive clone, and this is a positive clone. Like this, you can screen the cDNA library. What are the applications of cDNA library? When you're constructing a cDNA library, we have some without something you can do it with the cDNA library, right? Yes or no? Yeah. Here you can see it consists of cDNA of mRNA expressed in a particular conditions. So we can gain the knowledge of the genes responsible for particular condition. As I told you that a cell or organism, right, they express certain genes for certain conditions only. So when you isolate mRNA and convert it to cDNA and clone it and sequence it, then you will be knowing what are the genes which are particular to the particular condition. What are the genes which are expressed? What are the genes are responsible for a particular condition of the organism or particular phenotype of the organism, right? This can be done. And second point is that especially eukaryotic genes have introns and exons. When they are converted into mRNA and the cDNA, they have only exon sequence, not the introns. Introns are gone in the splicing. So eukaryotic cDNA can be cloned into the bacterial expression system, and you can get the large quantities of protein. Heterologous expression of is possible when you have a cDNA of eukaryotes. You can clone them into the bacterial expression system, and you can get the large quantities of protein. That protein can be used for the structural studies, raising antibodies, or therapeutic uses. Like you have many applications of that. Human insulin, produced in the bacteria. That's a recombinant insulin, right? Study of alternative splicing. One gene can encode the two mRNAs, right? So when you isolate mRNA and convert it to cDNA and you go for sequencing, then you'll be knowing a gene is able to go for two mRNAs. That is possible with the cDNA library construction. Yeah, cDNA library can be used to screen the homolog genes from other system. As I told you, if you construct the human cDNA library, that cDNA library can be used to screen the chimpanzee or gorilla. Right? Yes or no? Yeah. Yeah, we can identify and clone the novel genes which are expressed. Under every situation, there are certain genes which are highly upregulated, there are moderately upregulated, they are poorly upregulated, right? Some are novel genes are upregulated. So when you convert them to cDNA and clone them, there is a possibility to get the novel genes expressed under a particular condition, right? So these are the advantages of or applications of cDNA library. Same thing here. You see, I'm giving you an example here. Suppose you have a yeast cell culture, and it is when you expose to the high temperature, then the yeast cells, synthesize the mRNA, which are particular to the genes which are going to be renature the proteins of each cell. They're called as HSPs, heat shock proteins. You see, heat shock protein genes are expressed, and you get mRNAs. That mRNA are converted to the heat shock proteins. Simple thing is that when you expose yeast cell culture to high temperature, immediately they'll synthesize. The HSP genes are transcribed, and mRNAs are produced, and mRNA is converted into protein molecules. They are HSP70 or HSP90 or other HSPs. And what is their function is HSP70 or 90, restore the denatured proteins to the normal. Right? Denatured proteins to normal protein, restoring of function. Right? So this we can get through the cDNA. If you construct a cDNA library from these mRNAs, you will be knowing what are the genes are responsible for the tolerating or adapting to the particular condition. Right? And second one is eukaryotic protein expression, E. coli. This is a gene, and transcribed you got mRNA, and that is converted to cDNA. You have only cDNA have exons E1, E2, E3, E4. E stands for exon, I stands for intron, right? This cDNA is cloned into the PET system, PET vector, and the recombinant is this one. The recombinant is transformed into the E. coli, and you can use the IPTG or other inducer to express the gene of your interest, and you get large quantities of protein. This is possible only because you are converting mRNA to cDNA. You don't know the function of certain genes, but you have a cDNA copy, you clone it, and you test the function through the protein. As you can do, if you think that is an enzyme, you can do the enzyme assess. If it is a transporter, you can check the transport activity when you have a more protein, right? And study of alternative splicing. Study of alternative splicing. So one gene codes more than two mRNAs, you see. And their mRNA, this is a E1, E2, E3, E4. It is having all four exons. But other one is having only E1, E2, and E4. 3 is not there. So you can see, when you screen the cDNA library, there is a possibility to get the alternative splicing variants of the gene. These are the different, applications of cDNA library. Now, what are the disadvantages of regular cDNA library? Right? The main advantage is that, as I told you, highly expressed genes are more represented in the regular or normal cDNA library. As I told you that highly expressed genes will be there, moderately expressed genes will be there, and poorly expressed genes are there. When you take total RNA or mRNA, where one particular or few genes are highly expressed, and their mRNAs are counted to cDNA, and they have the good number, and the clones also will have a few genes cDNA molecule, not all the genes expressed under a particular condition. And poorly and rarely expressed genes may not be represented in the regular cDNA library. You can't, because always a big number, Suppose X gene is 1,000 copies. Y gene is only 10 copies. X gene is represented very well in the cDNA library, but Y gene cannot be because their number is less. Yeah, regular cDNA library is a partial representation of expressed genes under particular condition. Yeah. So because of the highly expressed genes or mRNAs and cDNAs are there, it is not going to be complete representation of the particular condition. It's a partial representation of the mRNA expressed in a particular condition. So these are the disadvantages of the regular cDNA libraries. This is all about today's class. Now we will go for a small assessment. Don't worry, they're not very tough questions, very simple questions. Yeah, you see, which enzyme has the ability to use mRNA and synthesize the cDNA? Options are Taq DNA polymerase, reverse transcriptase, primase, and the gyrase. Which one of the following is in vitro synthesized DNA? bDNA, aDNA, zDNA, and cDNA. During the first strand cDNA synthesis, the oligo-DT primer binding to which site in the mRNA? During the mRNA to cDNA conversion, you incubate the mRNA with the oligo-DT primers and DNTPs and reverse transcriptase. So the initiation of the synthesis all begins with when oligo-DT primer is bound with some part of mRNA. What is that? How do you generate cohesive ends on double-stranded cDNA to clone into a vector? As I told you that cDNA, which are double-stranded cDNA, do not have any cohesive ends. They're not compatible, directly compatible with the vector. So we have to add certain molecules to the cDNA. What are those? How do you screen an unknown cDNA library for a particular gene in a simple way? Suppose I said chimpanzee's cDNA library is constructed, right? Then how do you screen one or two genes out of it? So here we said let's if you are comfortable, you can answer them. If you are not comfortable, I'm going to reveal the answer also, no problem. The first, which enzyme has the ability to use the mRNA and synthesize the DNA? That's a reverse transcriptase. Which one of the following in vitro synthesized DNA is that's a cDNA is a in vitro synthesized DNA, not bDNA, aDNA, or zDNA. They are the different forms of the DNA. They're naturally present. Yeah. Oligo-DT primer bind to the poly-A tail of mRNA, right? Yeah. How do you generate cohesive ends on the double-strand DNA to clone into vector? You can use the adapters or linkers or all of them, right? Yeah. Here, fifth one is if you have a homologous gene, then you can screen simply, right? So this is all about cDNA library. definition and necessity of cDNA library, and the steps involved in the cDNA library, and types of cDNA libraries, right? So applications of the screening applications and disadvantages of the cDNA library. Thank you all for your patience. Hope you understand my lecture. If you have any questions and clarification, please get back to me through my YouTube channel. That's Dr. K PREM-PRIMER. And to get more updates, try to consider subscribing my channel, so that you will be knowing whenever I add new videos, right? So this is all for the day. Thank you all for listening, and see you again. Until then, bye-bye.